welcome back from your break, and we are now going to be looking at questions that can be asked on all aspects of plant structure and functioning. Questions often start off simply where you are given a picture. In this case, it's an example of a potometer, and you're asked, what does this piece of apparatus actually do? That's one of the straightforward ones, and of course, that tests the rate of transpiration. Then questions get a little bit more tricky, because if we look at this example, it shows us drawings of the surface of two leaves, and the dark things are the guard cells. And if you have a look, leaf A and B have a different distribution. Leaf B has got a lot more stomata, and you need to be able to say, okay, if it's got more stomata, it means it's going to be losing more water, all sorts of things like that. And questions will be asked to check to see have you picked that up. So if we have a look, it starts off with easiest questions. Okay, what is W? W is a pore, so it could be either C or D, and it's clearly the pore because it's pointing to the hole. So the next ones we need to look at, okay, what is X? X is a guard cell, so that is correct. So your correct answer there is C. Then, the next question now, this is testing your understanding, in what environment would this plant, leaf A, survive in? Okay, and if we look, it's got fewer stomata, so that tells us that it is adapted to a drier environment. If there are fewer stomata, it also means that less carbon dioxide can get in, so it would survive in an environment that has got a lot of carbon dioxide. So we can say, okay, it is drier, so that would work. Wetter, nope. Drier, yes. Wetter, nope. Then, would it survive if there was less carbon dioxide? Because it's already got less stomata. So if there was less carbon dioxide, it wouldn't be able to get in enough carbon dioxide. If it's got more carbon dioxide, it would be able to survive, so the correct answer there is C. Then, sometimes you're given photographs taken under a microscope, and you're asked to identify each different type of plant organ. So, let's have a look at this. It's a plant part taken under a microscope, and answer the questions. Is this a cross or a longitudinal section? And you should know cross-section is cut across like this. Longitudinal section is lengthways. And the only way you would get something like this is via a cross-section. Then, is it a root or a stem? And the key to that would be, look for the xylem. If the xylem is in separate vascular bundles, it's a stem. If the xylem is in the center, then it would be a root. And we can see clearly that there are separate vascular bundles, so that is obviously a stem. Then identify the layers. Okay, so let's have a look. Layer number 2, that's that. That's our bundle cap. Layer number 3, which is your phloem and cambium, and layer number 4, which would be the xylem. What is the function of layer 1? That's just underneath the epidermis, and that's where the parenchyma is, so its function would be support. Next one is a drawn picture of a leaf with a whole lot of the different parts labeled. Let's have a look. Okay, give the letter and name of the tissue responsible for transporting water. Okay, so you now need to look, and transporting water, that is T, the xylem. Transporting manufactured food away, that is Q, the phloem, and then the reason why it's 4 marks is because there's a mark for the letter and a mark for the name. Then, next thing you have to give the letter and name of the main photosynthetic tissue. And the main photosynthetic tissue here is R, which is the palisade mesophyll. Then it takes you further because you now have to describe 2 ways in which these cells are adapted for maximum photosynthesis. And that's for 4 marks. So you would have to talk about the fact that they are elongated, closely packed, few air spaces, filled with chloroplasts, directly underneath the upper epidermis. Any 4 adaptations which make them more effective at their job, which is maximum photosynthesis. Then, still on the diagram of the leaf, explain 2 functions of the cells labeled P. Okay, so P, that is the epidermal cells, so that would be protection. If they have a cuticle, it would be lessening of water loss. It could also be they're transparent to allow light in. Any 2 functions. Then, 4.5, name the cells labeled W and V. Okay, so V, that is those cells there, and W, that is your ordinary epidermal cells. And then you have to tabulate 2 structural differences. Okay, in other words, you need to say how are they different in structure. And obviously, if we look at the size, the guard cells, they're little cells. What is their shape like? They are bean-shaped. These cells are very often rectangular. What's different about these walls of the stomata? They've got thick inner walls and thin outer walls. These have got thicker outer walls and thin inner walls. Okay, so it's basically any 2 structural differences. The next one we're looking at is label S, and label S is pointing to the spongy mesophyll, and you have to give the primary function of this layer of cells, and because of the fact that there are so many air spaces, it's obvious that the function of this layer of cells is basically for gaseous exchange. Now, you will have noticed that as we've been going through the questions, gradually a little bit more has been incorporated because we started off with straightforward, what is this? Now we're bringing in all sorts of things, and this next question we're looking at is an example where we are also including different experiments that can be done, as well as the root, stem, and leaf, and the water movement through the leaf. Now, a question like this, to be able to do it properly, you really need to be able to understand and put everything together. So, let's have a look at this. Okay, we have a picture, and this is obviously, this is below the soil. On this side of the diagram, we have a picture of a plant with a root, the bottom here, below the soil surface. It shows root hairs, water moving into root hairs, moving up through the xylem. And B is obviously pointing to the stem because the water is moving up the xylem and it's going into the leaf. When the water gets into the small xylem vessels in the leaf, it leaves the xylem. Some goes into the cells, some goes out through the process of transpiration. Then, other pictures show different organs found at different parts of the plant, and an experiment. And the questions will need you to link everything together. So, always starts off with easy questions. Identify A, B, C, and D. A is the root hair, B is the xylem going all the way up, C is an epidermal cell, and D is your stoma. Then, describe the way in which part A is adapted. A is your root hair, so here again you are talking about an increase of surface area. Then, 3 adaptations of tissue B. And here's tissue B, and tissue B is your xylem. So you need to discuss 3 ways xylem is adapted, for example, no cross walls, thick walls to prevent collapse, no cell contents joined end to end, things like that. Any 3, because it's out of 3 marks. Then, describe the function of part D, and part D is the stoma, and you would simply say opens and closes to be able to let water out of the leaf or to let gases into the leaf. Okay, then it starts getting a bit more complicated because you now have to draw up a table comparing tissues E and F. The next question asks you to compare tissue E and F, and if we look at the picture here, tissue E is your palisade mesophyll and tissue F is your spongy mesophyll, and you have to compare them in terms of shape, round or rectangular, air spaces, present or absent, number of chloroplasts, lots or a little, and the main function, which is gaseous exchange or photosynthesis. Next question asks us to look at the process that occurs in part A, and here's part A, which is basically your root hair. Which process can be linked to the apparatus here? And the apparatus here is a diagram on osmosis. So answer to 5.6 is osmosis. Then if we look at 5.7, which part of the diagram 4, which is the bottom one here, corresponds to the following diagram 3, and your dialysis sheet corresponds to the cell membrane because they are both semi-permeable, and the sugar solution links to the vacuole, the water and the sugar in the vacuole. The next question looks at how much water different plants lose under different conditions. Okay, and there's a whole big explanation of an experiment in which a plant is placed in a test tube of water and it's placed in the sun for 5 hours. Then the same plant is put inside a plastic bag and placed also in the sun for 5 hours, but the plastic bag changes one environmental factor, which is humidity. Then the next question, 6.2, why is it better to use the same plant? And we've come across that before because different plants would have a different number of stomata. The next question looks at the graph of the results, and it shows that A loses a lot more water than in situation B when it's placed in a plastic bag. And what you have to do is explain how plant B loses a little bit of mass because it loses some water, but then the plastic bag stops it from losing any more water because the concentration or the humidity outside in the plastic bag would be very high. Then 6.4, what other conditions might affect the outcome and thus need to be controlled? And those are all the other factors that affect osmosis, which is temperature, light intensity, and wind. Then the last question, how would a farmer grow leafy vegetables, conserve water? And that would obviously be, he would put his plants in a greenhouse because that would keep the water that's being transpired, it would be kept in the greenhouse, it would increase the humidity so they lose less water. I hope that this revision lesson has helped you a little bit and you will feel more confident when you actually face questions that at first look quite difficult, but if you look at them step by step, you can get there. I hope this series of lessons has been of value to you. See you next time.